Hello, I am Dr. Gor Sarkisian. Welcome to BIO415 Molecular Biology. And today's lecture is going to be on cell chemistry and bioenergetics. And basically, in this chapter, we will go over the chemical components of the cell, the catalysis, and the use of energy in the cells, and how the energy is obtained from food. So here is the basic composition of a living system. Carbon, hydrogen, nitrogen, oxygen comprise 99% of the total number of atoms in a human body. The other 7 in the blue, which are phosphorus, chlorine, sulfur, sodium, potassium, calcium, magnesium, they constitute about 1%, less than 1%. And the green ones are the trace elements that are required for life. And the yellow ones have some role, but perhaps they're not nearly as essential as the other ones. So the living organisms are made of only 92 naturally occurring elements, with carbon, hydrogen, nitrogen, and oxygen making up over 96% of all of the organic weight. The atoms in the biological systems and generally are held together by covalent bonds to form molecules. The covalent bonds in the biological systems are hundreds of times stronger than the thermal energy within the biological systems, within a cell. So the covalent bonds in the molecules in cells cannot really spontaneously break. And they are broken only in very specific cases, only during specific chemical reactions that involve other atoms and other molecules. Even though the covalent bonds are some of the strongest, the molecules can be held together by much weaker non-covalent bonds. And in biological systems, these bonds are actually also very, very important. So here's a general outline of some of the common energies in a cell. So as we go towards the higher bonds of carbon-carbon bond, for example, we begin with average thermal energy, which is way less than a carbon-carbon bond. There is no way a carbon-carbon bond can deteriorate on its own in a cell. We have ATP hydrolysis in the cell, which is a very important event in energy processes in the cell. So this basically represents the bond strengths that need to be broken, right? And obviously non-covalent bonds are much weaker than covalent bonds. And it is represented in kilojoules per mole, so or kilocalories per mole. So if you remember from your physics class, a joule is an energy that is required to move an object 1 meter against a force of 1 Newton. Conversely, the calorie is the energy needed to raise a temperature of 1 gram of water by 1 degree Celsius. And these are basically the same measures in different systems. And they're interchangeable. 1 kilocalorie is 4.18 kilojoules. Plain and simple. So now let's talk about some of the bonds that exist, both covalent and non-covalent. Here is a table that shows bond types, covalent, non-covalent. And we have previously already established that the covalent bonds are strongest. And here you can see that we have covalent bonds of strength up to 377 kilojoules per mole in both vacuum and water. What does that mean? Well, if you take a graphene layer, for example, which is a chain of carbon-carbon bonds, if we submerge it in water or in a vacuum, those bonds between carbons are not going to change. And you'll see why this is important. Because if we look at the non-covalent bonds, such as bonds that are formed through electrostatic attractions, ionic bonds, first of all, their lengths are longer. They're about a bit less than twice as long as average covalent bond. Now, if you pay attention to the in vacuum strength, it actually rivals that of covalent bonds. It's quite strong. But in water, this bond strength goes down to 12 kilojoules per mole. It's deteriorated. Now, hydrogen bonds are weak bonds to begin with. And in a vacuum, they are slightly stronger. But obviously in the water, which is surrounded by hydrogen bonds all around, individual hydrogen bonds become less. And then the weakest form, but again, it's a weaker force, but a very essential force in all of chemistry. The van der Waals attractions. Even longer, 0.35 nanometers. And they are not even half a kilojoule per mole. But even then, they play significant role in biochemistry. And then another interaction that we have is hydrophobic forces, but they are obviously not listed here as bonds. They are not bonds, but they are very essential to biochemistry also. As we saw on the previous slide, the non-covalent bonds are just a fraction of covalent bonds. But when many non-covalent bonds come together and start interacting, they can form a very tight bonds when they're acted upon each other simultaneously. Now, this phenomenon involves all four non-covalent forces, which, if we remember, include electrostatic attractions, hydrophobic forces, hydrogen bonds, van der Waals attractions. Now, when we have macromolecules in a biological system, active proteins are actually monomers of macromolecules, dimers, for example. And these dimers could be either two identical proteins or two different proteins that come together to form a single functional unit. And they come together to form this single functional unit due to complementary non-covalent forces on both of the proteins that both allow association with each other, and more importantly, proper association with each other, which forms a functional protein. Now let's talk briefly about hydrogen bonds. Hydrogen bonds are roughly about 0.3 nanometers in length. They involve a donor and an acceptor atom. Donor atom is the one that has a hydrogen atom covalently bound to another one that is more electronegative, such as nitrogen in this case, which results in a dipole positive charge on the hydrogen. And on an acceptor side, the atom, the acceptor atom itself, is the more electronegative atom in that molecule, which has a partial negative charge on it. And the partial positive and negative charges between the hydrogen, the donor hydrogen, and the acceptor electronegative molecule result in weak interactions, which are known as hydrogen bonds. Review question 1. Please compare and contrast the carbon-carbon bond breakage energy and the complete glucose oxidation energy. And please tell me, why is this relationship important? Review question 2. Why are ionic bonds significantly reduced in strength when in interaction with water? Review question 3. What are van der Waals attractions? So now let's talk a little bit about acids and bases. So what are acids? In a nutshell, acids are molecules that can readily give away a proton, a hydrogen, positively charged hydrogen in water. So here is an example of adding acetic acid to water. Acetic acid has the OH group, in which the polarity is great. There is a great positive charge on hydrogen, and it can very easily give that hydrogen away to water. Once it interacts with a water molecule, we get a acetate ion, which is negatively charged, and we get a hydronium ion, which is positively charged. As you can see, it's an OH3. So effectively, this reaction adds a proton, a positively charged hydrogen, to the water solution. The image B on this slide shows how a molecule of water and a hydroxyl and hydronium ions are in equilibrium. Now, in pure water, there is always a concentration of hydronium and hydroxyl ions. And the hydronium ions are in equilibrium at 10 to the minus 7 molar. Now let's remember what the pH of pure water is. It is pH of 7. So by definition, the pH is logarithm of 1 over concentration of H3O plus. Okay, so now when we add acid to it, our concentration of hydronium ions goes up. Therefore, our pH drops, right? So we have a 10 to the minus 6 molar of hydronium ions now. So our pH is 6 now instead of 7, right? The bases now function almost exactly the same, but in the opposite to acids. For example, sodium hydroxide, NaOH, is a well-known base. When combined with water, it produces sodium ions and hydroxyl ions. Again, the pH is the equilibrium between hydronium and hydroxyl ions. And with addition of sodium hydroxide, we add hydroxyl groups. therefore skewing the ratio between hydroxyl and hydronium towards the hydroxyl. So in relationship, the amount, the concentration of hydronium ions drops, therefore the pH increases. Let's remember, pH is the logarithm of concentration of the inverse of the hydronium ions. Review question 1. What would happen if we add equal portions of hydrogen chloride and sodium hydroxide? How would the pH change and what would be the reaction products? Now that we have discussed what happens in the aqueous solution, in the cells, outside of the cells, let's talk about some of the organic components. Okay. Now, here are several basic functional groups that are prevalent everywhere in the biological systems. These are methyl, hydroxyl, carbonyl, carboxyl, phosphate, the amino, sulfhydryl, okay? And these guys are found everywhere in sugars, fats, nucleotides, amino acids, proteins. It is important to know these, so please learn them, memorize them. So here are the 4 main molecular families that comprise pretty much everything in a cell. And these are sugars, amino acids, fatty acids, nucleotides. Now, nucleotides are responsible for being polymerized into nucleic acids. Amino acids are polymerized into proteins. The fatty acids are basically put together into fats, lipids, and phospholipids for the membranes. And then we have sugars, which are polymerized into polysaccharides. Now, besides being polymers that are used in many different for many different functions, the sugars and fatty acids can also be energy storages. But these are not their only roles. The sugars and fatty acids can have structural, functional, signaling roles also. So to reiterate again, we have a subunit, we have a macromolecule. Subunit is a single unit that is put into a chain to create, let's say, a polysaccharide, if we put a chain of sugars together, or a chain of amino acids to make protein, nucleotides to make nucleic acids. Now, there is a well-defined chemical reaction that is a part of polymerization of a monomer into a macromolecule. And it is known as condensation reaction. Condensation reaction is basically a reaction that results in binding of 2 adjacent monomers, which has, one of them has an H group, the other one has an OH, and through this reaction a water is released and these 2 monomers are bound together. Now this reaction is energetically unfavorable because we have a pool of sugars, let's say, and then we make an orderly polysaccharide chain. So we need to input energy, perform work to be able to combine individual sugar molecules into a chain. That's just the way the nature is. It takes effort to clean out a room. Now the opposite also occurs constantly, which is a polymer being divided into its individual monomers. This reaction is called hydrolysis, in which a molecule of water is absorbed and is divided into H and OH, and subsequently we have the individual monomer that is being released with an OH group attached to it. This reaction, on the other hand, is energetically favorable and can occur without any energy input. So here is a typical condensation reaction. I hope you can recognize that this is a reaction between two amino acids. And we have, if you remember, we have the N-terminus and C-terminus of an amino acid, and it's the N-terminus interacting with C-terminus to form a polypeptide chain. So in this picture we have a COOH, which is the carboxyl group. The OH on the carboxyl group can be removed through a condensation reaction if it interacts with one of the hydrogens on the amino group on the adjacent amino acid. So through this condensation reaction, a bond is formed between the carbon and nitrogen of two adjacent amino acids, and as a result, a water molecule is released. Now, this reaction is not energetically favorable. It cannot happen spontaneously. It requires energy to be inputted into it, and we will definitely go in depth in what exactly that energy is and where it comes from. Now here is the opposite, the hydrolysis. And here we have the C double bond O single bond nitrogen, which is also bound to another hydrogen, the group in red. And the point of hydrolysis here is to break that CN bond. And that CN bond is broken when the carbon forms a new bond with the OH group on a water molecule, and the nitrogen forms a bond with the remaining hydrogen. And as a result, we have now two monomers of amino acids. And this reaction, on the other hand, is favorable, and it can happen spontaneously. So just to look at the overall composition of, let's say, a bacterial cell in this case, let's look at its chemical composition. A bacterial cell is 70% water, and the remaining 30%, 15% of that 30% is protein. We have a few percent of polysaccharides, RNA, DNA, and all kinds of small little ions there. We have small molecules, we have phospholipids. So this is basically the composition of a regular bacteria. So let's talk about the tertiary structures of polymers. So let's say in proteins especially, obviously the covalent bonds create the primary structure of the protein. But the important thing is that covalent bonds may have a degree of freedom, rotational and vibrational freedoms. Not always, but sometimes a single bond carbon-carbon or carbon-nitrogen, carbon-oxygen, they may have a certain degree of freedom. And so they can have some resonance conformation. But when the chain becomes long, the non-covalent interactions, which, if we remember, include hydrophobic interactions, ionic bonds, hydrogen bonds, these non-covalent interactions basically fix or limit the degree of freedom, and they help to fold the long primary chain of amino acids into a functional three-dimensional protein. So here we have a cartoon representation. Basically, a primary structure can have many different conformations, and many of these conformations are of higher energy, which means they're not stable, and through time they come to a stable folded conformation. I mean, that's partially because the inside of the protein is hydrophobic and it's trying to get away from the water molecules surrounding the protein. The protein might have some residues that attract each other because some have positive charge, the others have negative charges. And altogether as a system, they help to fold the protein properly, and the properly folded protein is biologically active protein. So here is an example of progression of a biological molecule from its individual constituents to a functional protein. For example, here we have sugars, amino acids, and nucleotides, which are polymerized into some globular proteins and some RNA, which are not combined together yet, so they're not functional. But then these molecules do aggregate due to these non-covalent interactions and form a functional protein, such as, in this case, a ribosome. The formation of the ribosome is done predominantly through non-covalent interactions, and now after the ribosome is complexed into a unit, it can be used to transcribe RNA into protein. Review question 1. Why the degrees of rotational and vibrational freedoms are important in carbon-carbon bonds when observing or talking about tertiary protein structure? Question number 2. Please explain how hydrophobic interactions are important in proper protein folding. Now, another very important molecule for biological systems are enzymes. And this is because chemical reactions that actually occur in the cell would normally require much higher temperatures to occur. And the cells require a tightly controlled mechanism to actually boost these reactions into happening. And these are controlled through a group of very specialized biological catalysts. And these are almost always proteins called enzymes, but sometimes RNA can also act as a catalyst, and these are known as ribozymes. The tight control of these chemical reactions happens because each enzyme is responsible for only one of many different types of reactions. And the catalytic process can be tied from one enzyme to another. because in this scenario, the substrate, or rather the product of one reaction, becomes the substrate for the subsequent reaction. As you can see in an illustration below, we start with molecule 1, catalyze a reaction through enzyme 1 to create molecule B, and then that molecule B becomes a substrate for enzyme 2 to generate C, and then so on and so forth. We all know that biological systems are complex, and we all know that many different things happen simultaneously. There are many different reactions that are seemingly opposite to each other but occur at the same time. So usually we have two opposite streams of chemical reactions occurring at any time. One is catabolic pathway, which is basically eating food, breaking it down to smaller molecules, harnessing the useful energy. Of course, some of it is lost as heat. And then basically breaking it down to small molecules that the cell can use to rebuild proteins that it needs. The other one is the anabolic pathway, or biosynthetic pathway. And this pathway basically gathers the small molecules that we generated in catabolic pathway and the energy that was harnessed through the catabolic pathway. And with those building blocks and the energy that we have now, we can synthesize new macromolecules. And if you remember a few slides before, we talked about the unfavorable reaction of forming polymers. So we need the building blocks as well as energy to rebuild new polymers that the cell needs. And these are the pathways that supply both the resources and the energy to do so. So as you can see, biological systems are quite complex and very organized. And even on the microscopic level, we can see how organized these structures are, such as viral proteins, the microtubules, in specifically in this case in a sperm tail. We have pollen. We have the cross-section of a fern stem, and we have a spiral arrangement of plant leaves. And if you'd like to learn more about that particular structure, it's please go and search for Fibonacci sequence. Just if you're interested, it's just fun. Anyway, so we will talk a little bit about entropy, which is the phenomenon of decline of orderness, and if no energy is put into the system will tend to become disorderly. Now here is an interesting picture. I'm not sure where this image was taken, but it looks so familiar. I think this happens at my house quite often also. So entropy is a fundamental law of physics. It's the second law of thermodynamics. It basically stipulates that in the universe or any other isolated system, the degree of disorder always increases. So basically the transfer from image 1 to image 2 is a spontaneous reaction, as time elapses, of course, right? But it's easy to let kids run around the room and then come back in half an hour and everything is destroyed. All the toys are everywhere, right? Easy. That's increase in entropy. Now, putting it back together, that requires time and effort and energy. But putting this in perspective, somehow the cells manage to remain highly orderly. It is true, however, that the entropy, the whole entropy, has to increase either way. That means something else has to become disorderly to keep the cells intact and orderly. Review question 1. Please describe the two pathways of metabolism. So here is an interesting model, but this model very clearly illustrates one way of how cells can remain orderly while not violating the laws of thermodynamics. So when we have a reaction that results in formation of a polypeptide chain, for example, that reaction results in generation of water and heat. So the idea here is that if that heat is dissipated to the surrounding, the overall entropy will increase, but the entropy inside the cell will actually drop. But if we take the whole system as a unit, we have an increase in entropy. So we are not in violation of the laws of physics, thankfully. So I think this is a nice and clever way to illustrate it. Of course, the reality is a lot harsher than what it is, but it's actually quite a good way to show it. So the cells obtain their energy by oxidization of organic molecules. Okay. For example, both plants and animals harvest the energy from organic molecules, right? The sugars, for example, that the plant has photosynthesized as food for itself, and in terms of animals, the mixture of large and small molecules that animal has eaten. In both cases, the energy is extracted from food molecules by a process of gradual oxidation. Now, oxidation is a very interesting reaction. It's basically burning. An explosion of TNT is also oxidation. Burning wood is also oxidation. Now, we know both of them result in release of extraordinary amount of energy. In biological systems, a gradual oxidation means that we can actually harvest that energy to actually do good. So cells obtain energy by oxidizing organic compounds, organic molecules. So we have two systems here. We have photosynthesis and cellular respiration. Photosynthesis is a process in which carbon dioxide and water are taken up, and the reaction is taken place with when an energy from light is used to produce oxygen and sugars. So this reaction happens in plants, in algae, in some photosynthetic bacteria, and it requires energy from the sun. Then, when animals eat plants, animals uptake sugars, and then that sugar is burnt in a controlled manner. Oxidation is a combination of a molecule that has potential to burn and oxygen, right? So we take sugars and combine it with oxygen. We harvest useful energy, and we generate water and carbon dioxide. And that process is known as aerobic respiration. So basically all living things, plants, animals, microorganisms, are a part of each other's environment, so they rely on each other. For example, plants photosynthesize. They take the carbon dioxide from the environment and produce sugars. When plants, algae die, they produce sediments and will decompose into fossil fuels. They can produce humus for plants, for new plants to be nourished. The plants are food for animals. Animals also, when they go through what we've discussed in a previous slide, the aerobic respiration, they generate carbon dioxide, which refuels the environment for the plants again. And that chain of reactions basically continues over and over. And it's usually in balance, unless it's no longer in balance because of, well, I don't want to get into politics, but shall we say special interests that may or may not drive the process of disbalance in our environment. So oxidation is burning or rust. But what does it really mean? Oxidation and reduction, which is the opposite of oxidation, involve electron transfer. In an oxidation reaction, an electron is removed. In a reduction reaction, an electron is added. So whenever we have an oxidation or reduction reaction, we always have both. Okay, so one compound is oxidized, whereas the other one is reduced. For example, ferrum 2+ can be oxidized to ferrum 3+. Chlorine can be reduced to chlorine-. Does that make sense? If you have any questions on this, please comment. Review question 1. Please briefly describe oxidation in cellular respiration. So to illustrate this better, oxidation and reduction can also happen between the members of a single covalent bond. So in this case, for example, we have the atom 1, atom 2, and which form a polar covalent bond. Polar covalent means partial negative and partial positive, right? Now, excuse me. The atom that has a partial positive charge is oxidized, and the one that has a partial negative charge is reduced. Well, in terms of organic molecules, it's very easy to spot oxidation and reduction. Okay, so let's say we take the simplest form, methane. Methane has 1 carbon and 4 hydrogens attached to it. Okay, so the methanol is basically oxidized methane, and it is the bond, the number of hydrogen bonds to carbon. So carbon dioxide is the most oxidized form of carbon in comparison to methane. Now, interesting thing is, if you think about it, methane is highly flammable and carbon dioxide is not flammable at all. So oxidation in this case, it's very easy. You light it up, carbon dioxide is generated, and we have heat generated. But the reduction reaction is not as easy. It requires input of energy for it to occur. So what does it really mean that it needs energy to occur? Okay, well, again, just like on a previous slide, if we take paper in oxygen, light it up, we'll have smoke, ash, heat, carbon dioxide, and water forming. Okay? So the paper does not really spontaneously combust, right? But if we give it some push with a little bit of a match, let's say, then this reaction will happen spontaneously from that point on. Why? Because it's energetically favorable reaction. It results in increase in entropy. We had one ordered piece of paper, now we have ash, we have smoke, we have heat, carbon dioxide, we have water, right? So the amount of disorderedness in the world has increased. But if you look at this energy diagram, we have to go through that hump from B to A for the reaction to occur, and that's bringing a lit match to the paper. It has to go through it for the reaction to occur, and once it passes over that hump, the reaction will proceed. Of course, in biological systems, we can't afford to light a match, right? That's why we have enzymes to help reduce that hump for us, and it can reduce the that hump significantly. It can increase the reaction speed by factor of 10 to the 14. Therefore, we can have some complex chemical reactions in the cells without burning the whole place down. The reactions are actually catalyzed. The activation energy is lowered. Review question 1. In this slide, we have a reactant Y undergoing a chemical reaction to produce a product X. Please tell me if the opposite of this reaction is possible and justify your answer. So we talked briefly already about how enzymes help to drive chemical reactions to a desired finish, but let's look at it from a different perspective. Now we have substrate and we need to get the product, and there are many different reactions that can happen to get to many different products from that one substrate. But here we have, let's say, that substrate can be divided into, or it could undergo 3 different reactions. On this diagram, on the right, we have the 3 branches when the substrate approaches the crossroads there. And now we have 3 different enzymes, and one of the enzymes is at high concentrations. So the likelihood of that high concentration enzyme to interact with the substrate is much greater than that of enzymes of lower concentration that will have a different product. And the same thing stands true throughout this whole pathway. The cell regulates the concentrations of enzymes that drive the substrate to the appropriate product. This is just one of the mechanisms through which a cell can drive chemical reactions to their favorable finish or appropriate finish for the cell. So we talked about entropy, we talked about disorderedness, we talked about some chemical reactions occurring spontaneously, some not occurring spontaneously. So let's look at the basically the physics behind it and determine what is it exactly that determines which reaction will proceed spontaneously. So let's consider a conversion of Y to X, okay? So the free energy of y is greater than free energy of x. Therefore, the free energy change, the delta G, it's universally known as free energy change. The free energy change delta G is less than 0. So let's look at the analogy of a spring to visualize what a negative delta G is, or a energetically favored reaction is. We have a compressed spring, and when we release it, it expands, releasing it energy. And if there was a pebble in front of it, it would throw the pebble across the room, and in so doing, it releases heat to its surrounding, right? So it was more orderly, it was more compact. Now when we released it, it became disorderly, it released heat, it increased the disorderness of the world. So same thing happens in a chemical reaction. When we have a negative delta G, we have a substrate, which when it undergoes a chemical reaction, it creates a product that is of lesser energy. So delta G is negative. Same thing is true with when one dissolves sugar in water or salt in water. Salt is a crystal, and when it's dissolved in water, it becomes the crystal structure deteriorates, and the entropy increases. Now, when the delta G is positive, that reaction is unfavorable. It's synonymous to that of compressing the spring back to a compressed form. So this kind of reaction is the joining of amino acids to form a polypeptide chain. Such reaction can only occur when it is coupled with a secondary, more energetically favorable reaction. So what does that look like? Review question. Please describe what happens during hydrolysis. So here is an illustration of coupling of two reactions. The reaction of C to D and reaction of X to Y. Now X to Y, it goes up, so delta G is positive. The C to D goes downwards, the delta G is negative. So the reaction C to D will occur spontaneously, and while doing that, it will drive the X to Y reaction with itself. Okay, and we will talk about how this happens in amino acid peptide formation later in the class. Well, thank you very much for watching the presentation. I hope you were able to find at least 10 questions to answer. May I remind you that the questions are both in a written and in a verbal forms. So please answer the questions and send them to me via email. And these questions are a part of your participation as well as assignment work. Thank you. Question 1. In a few words, please describe what reaction activation energy is. Question 2. If you take our universe as a single system, what is happening with the entropy within the system? And what does that mean in the long run?